Okay, let's move on. Right, so exams, I'm hoping to have the grades posted by the end of today, by Monday at the latest, well, which really means sometime over the weekend. And I'll give them back on Monday. Sorry, I was pushing for today, but unfortunately I have other things that I have to do here other than teach this class and grade exams. I wish that's all I had to do, because then I'd be on it. Um, Recitation for next week is not a paper, it's practice problems. So when you go to look, you'll see it's a folder that's empty, and you'll want to email me and say, where's the paper? And I'll say to you, it's not there because there's no paper. And so by Sunday night, I'll have posted practice problems that you guys will go through, and they'll be based on the signaling stuff that we're doing, and they'll hopefully help you kind of figure out how to think about the questions I asked on exams. Yes.
Are we doing the practice problems in recitation?
You're doing the practice problems in recitation in groups, and there will be a quiz. So it's perfect, right? Because you work through the problems and then I quiz you. Really can't go wrong there, right? If you are one of these people who just doesn't do anything in recitation, which is there's very few of you. Ooh, someone changed their hair color back there. Sorry. Then you're gonna be in trouble. But should be good. That's business. Okay, so far we've been talking about signal transduction and the major concept, which I'm really trying to drive home, which is that it's just a way to relay a message from outside the cell to the nucleus to affect gene expression or to affect cell function in some kind of way. Big picture. So when you start to get bogged down with this protein binds to this protein, always take a step back. There are three kinds of receptors that I introduced, and we're going to start going through them today. GPCRs, which the Nobel Prize was won for the other day, enzyme coupled and ion channel coupled. And then we talked about the concepts of domains within proteins, the business ends of different proteins, and molecular switches, GTP binding proteins, and proteins whose function are affected by their phosphorylation status. Yes. One last thing about the test. If you did not so good, if you weren't happy with your performance, please come see me. I really want to make sure that we could figure out how you can approach my questions so you could do better next time. Anyways, okay, so don't lose sight of the big picture. Please do not lose sight of the big picture. I've seen that a lot in recitation this week, where people are talking about beta-catenin pathway, Wnt signaling pathway, and beta-catenin, and I'll say, well, what's the function of Wnt signaling? And people will say, well, to do something with GSK3. And I'm like, that has nothing to do with no, that's not the big picture. And then so once we step back and we realize, oh, it's involved in signaling, involved with gene expression, involved with development and tissue regeneration and stem cells, and when things go wrong can result in cancer. Big picture. And that should help you to understand this, because we're going to dive into details now, and I want you to be able to back up and place it somewhere in the main idea of what's going on. Right, so the big picture is an extracellular signaling molecule coming from outside of the cell somewhere, from another cell probably. It binds to some kind of receptor. We just listed a bunch of kinds of receptors. And then something happens inside, which results in either a gene regulatory protein going to the nucleus and altering gene expression. There could be other enzymes being activated. There could be cytoskeletal proteins that are changed in some way. And once we talk about cytoskeleton and cell movement, we'll see how that ties in. But signal from outside the cell affecting cell function. Okay, so GPCRs. We started talking about that. These are these G protein coupled receptors. So it's a receptor that's usually 7 or multi-transmembrane domains, and some kind of G protein, which isn't in this picture, that's going to associate with it and regulate it. So there are many different types of G proteins. It's not many types of GPCRs. It's not one receptor. It's a class of receptors. And they all have a similar structure, and they all use G proteins. So that's why they're called that. Oh, I'm tired today. Who's tired? Yeah. Are we happy it's Friday? Mm. Ah. Okay, so this is the figure I showed you the other day from the Nobel Prize folks. Right, so here's your receptor here, and your hormone or whatever signaling molecule binds. And there's your G protein. And it's going to bind and become activated, and then they're going to go off and affect the cell's function in some way. And then another G protein can come along and become activated and go off and affect the cell's function in some way. So I said they're GTP binding proteins, right? So that means that they can be GDP bound and they're off, or GTP bound and they're on, and so they have an intrinsic GTPase activity. What does GTPase activity mean? Thank you. Hydrolyzes GTP. ATPase, what does that mean? So if you hydrolyze GTP, what do you get on the other end? GDP. Okay, right. So that's GTP to GDP. So when you're inactive, you're GDP bound, and when you're active, you're GTP bound. This conformational change within this protein allows for interaction with a target protein of some sort. So you'll have a signaling molecule that will bind to a receptor, which will activate a G protein, and that G protein can then go interact with something else and activate it. And that's how the signaling begins. Okay. So here's how it can look. So you'll have a trimeric GTP binding protein. What's trimeric mean? 3. Right, so it's a protein of 3 subunits. Alpha, beta, gamma subunits. And it's going to mediate interaction between a receptor and its target protein. So here's your receptor, and it's inactive. Here's your G protein. Notice it's membrane bound, and it's inactive. And here's some kind of enzyme that's inactive. So this is how it looks before a signaling molecule binds, right? So once the signaling molecule binds, which is right there, notice your G protein kind of moves over, becomes activated. And then the activated G protein can then interact with the enzyme. And it activates the enzyme. And then the enzyme could do something. What's that enzyme likely to be doing? In very broad general terms. What effect will it have on signaling? Will it promote it or inhibit it? If you're activating a signaling pathway. You probably promote it. I know, I tend to be really broad and people get confused because they want to give me protein names. So this is one way of we've seen a lot of relaying a signal downwards. Well, this kind of like starts off going across the plasma membrane, but eventually it's going to go downwards towards the nucleus. So it mediates an interaction between the receptor and its target protein, which is usually a plasma membrane-bound enzyme or ion channel. Okay. So GPCR activation, that means the receptor, when it's bound by its ligand, it induces the release of GDP by the G protein, which allows GTP to bind. Should start to sound familiar to people who've thought about G proteins. We've dealt with a fair number of G proteins already. Can someone give me examples of G proteins that we, bless you. Oh my Lord. So what other G proteins have we dealt with already? Well, the import and export in themselves are not G proteins, but you're in the right process.
So like RAN.
RAN, right? Remember RAN, GTP, RAN, GDP. What other G proteins have we seen? Huh? RAB. I hear someone saying RAB. RAB. right? And remember, in all of these cases, when it's GTP bound, is it on or off? And when it's GDP bound, it's off. And by on, it means it can associate with something, right? And usually activate something, right? If you start to see themes in things, A, it might be an exam question, and B, it helps you to understand things. Because if you can see in all of these situations, things act similarly, you can understand what things do, right? Maybe. Okay. So signaling is active until GTP is hydrolyzed, right? So you activate the GPCR, and then the G protein can go on, right? So you have GTP bound. G protein can go on and activate other things, and it's going to keep doing that until GTP is hydrolyzed. And GTP hydrolysis is regulated by its own, or it's stimulated by its own GTPase activity. So it's alpha subunit, which I don't remember which one that is, probably this one, because it's binding GDP, is going to stimulate hydrolysis of the GTP to turn it off. And there's, of course, whenever you think about things being turned on and off, you have to ask yourself how this is regulated, right? Is it random? Are G proteins randomly turned on and off? No. Would it make any sense for them to be randomly turned on and off? No, because that would be saying signaling is random. And gene expression is random, and cells function in random ways. Cracking myself up this morning. Can you imagine an organism where everything's just random? It's like, hey, we're going to make a placenta. We're male. And we're still going to make a placenta. Anyways. Just start to think in like these broad terms, and you're going to realize regulation is really important. Oh, God. I need to put a couch up here so I can lie down and relax. Okay.
Somebody did go out.
Oh, really? Did I let you out? Okay. I thought I was going to need a note from your mom. Okay. Here's you're going to be like, Dr. Ehrlich was on crack today. You know what it is? It's the Starbucks. Whenever I have normal coffee, I'm okay. Starbucks, it's like they pour Red Bull in there. Okay, regulation of GTP hydrolysis. So this can be regulated by binding to the target protein, right? That's the enzyme that's being activated, or some other protein, which are those other proteins are called regulators of G protein signaling. Makes sense. RGS. It makes sense that you might activate hydrolysis of GTP, right? Activate your GTPase activity when you bind your enzyme, because once you bind it and activate it, maybe you should turn off. Yeah, maybe. Okay, so in this situation, one protein is acting as a gap and another is acting as a GEF, right? So what's acting as a gap? So now you got to go gap stimulates hydrolysis. What stimulates GTP hydrolysis in this situation? A GTPase. Thank you. What stimulates activity of the GTPase? Anyone? Who's here? Anyone here today? Okay. Okay, so I just told you about how okay, well, let's go through the picture. Okay? So you've got your inactive GPCR and you've got your inactive G protein, and a signaling molecule is going to combine, and that's going to result in activation of your GPCR. So you'll get exchange of GDP for GTP. Would that be a GAP or a GEF? GEF, right? Exchange. So your GEF would be basically binding of your receptor by your signaling molecule. And then I just said GTP hydrolysis is regulated by binding to the target protein or another regulator of G protein signaling. Hydrolysis is stimulated by a gap, right? So this is similar in how it's working. There might not be RAN-GAP and RAN-GAF, but there are proteins with those functions, Remember, RAN-GAP and RAN-GEF were all about regulating the activity of RAN, right? And if you think about RAB, we didn't talk too much about RAB, but RAB is also regulated by a RAB gap, like a gap and a GAF also. Something's got to regulate if it's GTP bound or GDP bound, right? To keep it from turning on when it shouldn't be on. So something to think about. Okay, does anyone wonder what this is all about? Are you guys wondering why I'm going on and on about gaps and gaps? No, everyone feels good? Everyone's ready to take an exam? I'm going to give you the exam early. We're going to do it right now. Yeah, you guys ready? Okay. No. Okay, good. I want honesty. Thank you. Okay, so I'm telling you all this stuff about receptors and G proteins and activating things. And you're saying, okay, well, you keep talking about big picture. What's the big picture? Okay, so what is the GPCR doing? We're talking about activating it. So what? Well, sometimes it helps to think about things in terms of what is the actual outcome. So some G proteins are going to regulate cyclic AMP production. And cyclic AMP is a small intracellular signaling mediator. Woo, that sounds really scientific. It's a small molecule that activates other molecules that are involved in signaling. Okay? So an extracellular signal will bind to a GPCR, will activate a G protein, and it will activate adenyl cyclase, right? And you'll get an increase in cyclic AMP production, which puts all these signaling molecules in the cell to activate other things and pass the signal along. So it'll stimulate a downstream response. And some examples of signaling molecules that work this way are adrenaline, adrenocorticotropic hormone, glucagon, thyroid stimulating hormone. A lot of hormones work this way, and also a lot of signaling involved in more like physiologic responses work this way. So let's see a picture that helps. Well, the other thing to think about also is that G proteins, they could be stimulating or inhibitory. So there's one G protein, that G sub S, the stimulating one, that activates adenyl cyclase, which increases cyclic AMP. And then there's an inhibitory version of the G protein, which will inhibit adenyl cyclase. And it does this by regulating ion channels. Okay, so what am I talking about? I'm saying that there's all this cyclic AMP, and that's important in signaling. Well, cyclic AMP, it activates cyclic AMP-dependent protein kinase. It activates a kinase, right? And it's called PKA, and that relays a message to other proteins through phosphorylation. So you'll have your inactive PKA, so kinase, and when cyclic AMP comes along, it'll bind to it, and it kicks off the active subunits. And so now it can go on and activate other things. Okay, so what's this have to do with anything? So here, signaling through a GPR. This is an example. This is not all GPCRs do. It activates adenyl cyclase. So here's your signaling molecule, your hormone or whatever you want it to be. And it binds to your receptor, which activates your G protein. Right here you see G2P bound. And then interacts with this other enzyme, which in this case is adenyl cyclase. And then you have production of cyclic AMP, which goes on and it binds to this inactive PKA to activate it. And then the active PKA goes into the nucleus and it activates gene expression. Do you guys see a theme in signaling? Receptor, activation of things, go into nucleus, activate gene expression, right? So increases cyclic AMP, activates a kinase, goes to nucleus, activate gene expression. Big picture, yes?
To be clear, you're saying that cyclic AMP is just an example and G proteins act the same way on the hormones listed?
The hormone would be what binds on the outside.
Okay. So it always increases.
No, cyclic AMP is an example also. Yeah, so this one thing is an example. But you could also, well, we'll go on to other things, but one way to think about GPCRs is they tend to increase the concentration of small intracellular mediators. So small molecules, like cyclic AMP is really small. I didn't put the structure, but you guys, it's really small compared to like a protein, right? And another one is calcium. You guys have heard about calcium regulating things, right? So, so here's an example with cyclic AMP. So serotonin, neurotransmitter, acts through GPCR to increase intracellular cyclic AMP. Let's turn off the light. Okay, so this is a cell. What kind of cell do you think that is? What does it look like? A neuron. Okay, we'll go with that. Okay, so this is time 0 and this is 20 seconds. And here they add serotonin. So at 20 seconds after they add serotonin, and what they're looking for is concentration of cyclic AMP. Okay, so time 0, blue is very little and red is a lot. So at time 0, not much cyclic AMP. They add their serotonin, it binds to the receptor, activates G protein, activates adenyl cyclase, increased cyclic AMP, all in 20 seconds. And look at that. And so you have increasing amounts of cyclic AMP. Where do you think your receptor is located? If red is a lot and blue is a little? Probably somewhere near the red. Would be logical. So that's what it looks like. Okay. So lots of cyclic AMP, and I just told you cyclic AMP activates a kinase, which then goes into the nucleus to activate gene expression. So are you guys starting to see the big picture here? I see a few nods. Who really doesn't know why they're here and is wondering if withdrawal, like what the withdrawal date is? Okay. Okay, so G proteins are also targets for bacterial toxins. So if you've ever wondered how cholera toxin or pertussis toxin works, here we go. Okay, so cholera toxin. Okay, let's orient ourselves. I always get turned around in this figure here because I have to find a better figure. Okay, so here you have your G protein. And it's GDP bound, so it's off, right? It cannot activate adenyl cyclase, right? And so normally you'll get a hormone that will bind to a receptor, and you'll get GTP. You'll get that exchange, and GTP will bind, and you'll have activation of your G protein. Now, when you have cholera toxin, it actually ADP ribosylates your alpha subunit of your stimulating GTP, which basically keeps G protein, which basically keeps it on. It keeps it so you can't. It basically it's like throwing a wrench into the gears of the GTPase activity, and so it inhibits GTP hydrolysis, and you have this constant production of adenyl cyclase, and that. In intestinal epithelial cells, because remember, what does cholera cause? Diarrhea. What's diarrhea? I'll put that on the exam. If you ever, what did I have to take? In grad school, I had to take parasitology, and there was one guy who works on Cryptosporidium. I don't know if you guys know what it is, but it's a parasite that gives you diarrhea for a really long time. And he actually brought to lecture diarrhea. Like he brought like a huge bottle of it. I was like, I guess that's a way to get a class's attention, right? I'll try that next time. Next time my kids are sick. Okay. So that results in that results in intestinal cells. In this situation, results in efflux of chloride ions and water into the gut. When you have lots of water in your gut, it's not being reabsorbed. That results in diarrhea. So that's how cholera toxin works. Okay, and then there's pertussis toxin. Who here has been revaccinated? Okay, good for pertussis. That's an important thing to do, especially if you're around little kids. Okay, because you know that immunity is not it's not long lasting. Okay, anyways, pertussis toxin, it does the other thing. So, cholera toxin, it basically keeps it on, so it keeps the stimulating version of the G protein on. So you're getting lots of cyclic AMP. And pertussis toxin, it catalyzes ADP ribosylation of the inhibitory G protein. And so it prevents interaction with receptors and it prevents it from turning on. So it keeps the G protein off. And that's how it works. And I don't have a nice. Okay, doesn't matter. Okay, but these toxins are actually useful lab tools, okay, for examining whether a GPCR dependent response is mediated by a activating G protein or inhibitory G protein. And I think that will be a really great practice problem for us to talk about next week in recitation. Okay, so other roles for GPCRs. Cyclic AMP, just one example. People get really hung up on examples that I give as the way, and I want you to remember, it's one example of a GPCR mediated signaling pathway. Another one is activation of phospholipase C beta. This is a plasma membrane bound enzyme, so it's on the inside of the membrane, of the plasma membrane, and it regulates a whole bunch of processes such as glycogen breakdown, amylase secretion, muscle contraction, through IP3 or PKC induced effects on intracellular calcium. Remember, I was talking about calcium. You guys have learned about the smooth ER, rough and smooth ER, and it's the smooth ER is a storage for calcium, which can be released, and that regulates certain processes. So here's where we're going. The other thing that GPCRs are involved with is regulating ion channels in the plasma membrane, either directly or indirectly. And I don't think we're talking too much about that. Okay, so what am I talking about with phospholipase C, and what is she saying? So here's your GPCR, right? Here's your G protein. Here's your phospholipase 3. Your phospholipase C. So remember, in all these situations, everything's up near the plasma membrane, right? That's something important to kind of keep in mind. And then things are moving on. So if I ever ask you on an exam something about some enzyme and where it might be located, or just keep things in mind. And when you're drawing out processes, this is all up near the plasma membrane. Okay, so phospholipase C is activated, and then there's other kinases or other molecules involved, which then activate a kinase. Here it is, kinase, PKC, which then opens up a gated calcium release channel in the ER. And so lets all the calcium out. And what's that calcium going to do? It's going to go activate kinases and other things and result in just effects on the cell, gene expression, or other functions. Keep in mind, cyclic AMP and calcium are intracellular mediators. So when you think GPCR, think intracellular mediators. These small molecules, is calcium small? Is calcium smaller than a protein? Okay. And so is cyclic AMP. So these small intracellular mediators, they activate other enzymes that relay messages that affect gene expression or other effects on cell function. Okay. You're doing good today. Okay. So smell and vision are regulated by GPCRs that regulate cyclic nucleotide gated ion channels. Cyclic nucleotide. So what I say is, so cyclic AMP is a cyclic nucleotide, right? Because AMP, right? And so that's what it looks like. So this is GMP, right? Guanine. Cyclic AMP would have an adenine right there. So when I say cyclic nucleotide, it's accounting for both cyclic AMP and cyclic GMP or other kinds of nucleotides. Okay, so let's talk about the olfactory receptors. So they are GPCRs that bind odorants and act through cyclic AMP. So you've got your olfactory neurons, right? And so you'll have an increase in cyclic AMP when the odorant binds the receptor, and that results in CAMP-gated cation channels opening, allowing sodium ions in. You get depolarization of olfactory receptor neurons, and that sends a nerve impulse to the brain, and then you smell something. So just an example of something else, not just gene expression, but something else that's regulated by GPCRs. And here we're dealing with cyclic AMP gated ion channels. Okay, so we're talking about turning things on a lot. Don't forget, we need to turn things off. Right? You have anything you turn on needs to eventually be turned off. So you can have receptor desensitization after the exposure to ligand. You've seen this before, right? So you can have endocytosis. You can have you can have degradation, right? So you can either sequester your receptor, you can actually send it to the lysosome and you can degrade it, you can inactivate it by having some kind of protein bind to the cytoplasmic domain of your receptor. And then there's some other things also. Okay, so desensitization of GPCRs would fall into the category of this one right here, right? So you get phosphorylation, and then you get binding of the receptor by a protein called arrestin. That's an easy one to remember, because arrestin, what does it do? Does it activate things? Probably not. So you have your activated receptor, and then you'll get phosphorylation by a GPCR kinase, and that will result in creating a binding site for arrestin. So whenever you have phosphorylation, you're usually creating binding sites for recruitment of other proteins. And so if you have arrestin bound right here, can you interact at all with your G protein? Okay, thank you. I see some nods. Just think about it, right? What does it normally look like? So it normally looks like this, right? You have this interaction, right here. Here's your G protein. I mean your GPCR and your G protein. That interaction between your receptor and your G protein is what activates it and turns it on and allows that exchange. That's acting as your GAF, exchanging your GDP for your GTP, right? Activating. Okay, let's go back to the arrestin. So now that you're phosphorylated here and you recruit arrestin, you can't activate your G protein anymore. So that's a way to shut off signaling. Okay, so things to think about. How does the kinase know when to desensitize the cell? There's got to be some sort of mechanism there. Actually, let's make this. I don't have the answers here. Let's make this an extra credit because I'm feeling bad after grading exams. And it's all in the lecture, okay? So no one has any excuse for not answering the question properly. So how does the kinase know when to desensitize the cell? Or I guess the receptor in a way, right? What happens if you have a constitutively active GPCR kinase? What is arrestin doing? What would be the effect of a mutated arrestin? These are all good ways to think about things, right? What is the role of arrestin in endocytosis? Ooh, right? Because, well, we'll talk about that. And what might be the fate of an internalized GPCR arrestin complex? Yes. Yeah. I think that's a really great way to think about this. Yeah.
You have this online, or should we write it all down?
Oh, I post my lectures. Yeah.
Okay.
Yeah.
Okay.
Yeah. So this will be posted. So you guys have until, I guess, this about tomorrow sometime, anytime tomorrow, to get it to me for extra credit. And you've got to answer them all. So do one by one. Don't just like give me some random answer to one part of it. Yes.
We don't have to get a separate article, right?
No.
We
You
can just use them.
you can just use the textbook. Yeah.
So anytime tomorrow?
Anytime tomorrow is fine. Yeah, it could be anytime tomorrow. Okay. Because I won't get to look at it until tomorrow night anyways. Okay? That's a point. Okay. Everyone feeling good? Okay, so let's move away from GPCRs. Is everyone tired of GPCRs? Okay. Well, you're going to have to revisit them at some point when you study. Don't get too tired of them. Okay, signaling through enzyme-linked/coupled receptors. The reason it says linked/coupled is because that's two categories of enzyme. I'm sorry. That's two ways that they're referred to in different textbooks. Sometimes they're called enzyme linked and sometimes they're called enzyme coupled. We'll talk about it. Okay, so there are 6 classes. There are receptor tyrosine kinases, tyrosine kinase associated receptors, receptor serine threonine kinases, histidine kinase associated receptors, and onwards. Don't memorize this. There are 2 main classes. Okay. There are 2 main categories. One is our receptors that have intrinsic kinase activity. What does that mean? No. Yes. It can phosphorylate things. What does intrinsic mean?
It does it itself.
It does it itself. Right? So there are kinases that have intrinsic kinases. There are receptors that have intrinsic kinase activity. Yeah, they phosphorylate things themselves. They have their own kinase domain. Where might that be located if you're a receptor and you were involved in signaling? Would it be in the extracellular domain, the transmembrane domain, or the cytoplasmic tail? Okay, good. Cytoplasmic tail. So you don't even need study. And then the other category, so those are ones with intrinsic kinase activity. The other category are kinase associated. So here there are a bunch of different kinds, but they kind of all fall into those 2 categories. Kinase associated receptors. So what might be the difference between a receptor with intrinsic kinase activity and a kinase associated receptor? Just based on the words.
Can't the latter only associate with proteins that have already been phosphorylated?
Nope. Nope. Let's keep going. Someone else has got an idea. So we talked about intrinsic. I mean, you might be right in a way, but that's not the defining. You're not wrong, but that's not the defining. Yeah.
Maybe that the part that is with the kind of activity is bind to the.
Right, right. So one would be. So one would have intrinsic kinase, K, activity. And then the other would bind to proteins, separate proteins with kinase, which are kinases with kinase activity. Kinases with kinase activity. So this would be, so an example is receptor tyrosine kinase has intrinsic kinase activity, and then these associated receptors. So kinase associated. So those are two classes that we'll be talking about. And I just want you to understand the difference between them. So one has one protein, one has two. Kind of think of it that way. Any questions about the difference? Yes.
What was the significance of the distinction?
It's just that we're going to go through different signaling pathways, and they're different in how they function. Yeah. But I know that people always get confused about the difference, and so I'm putting it out there right in the beginning. Okay. So receptor tyrosine kinase subfamilies, they all have common domains, which is why they're all receptor tyrosine kinases. They have some kind of outside domains that bind something. They'll have some kind of transmembrane domain. But most importantly, they all have a kinase domain. I think this might be my last slide. Yep. I'm short today. Before we end, does anyone have any final questions? Uh-oh. Everyone's ready to leave. Okay.